We are going to start the new topic, and that is the sex determination. So in 10th class you have learned the basic thing regarding the human sex determination, and the topic was regarding whether it's the X carrying or the Y carrying sperm which fertilizes the egg, because egg will always have X and sperm can carry either X or Y. So now we are going to learn this concept in details of different methods as well as in different organism. So the first method of sex determination was proposed by Wilson and Stevens in 1902 to 105, and that mechanism is the sex chromosome mechanism of controlling the sex determination. That means there is a chromosome which controls the sex. Now we all know the background now very well, that the chromosome which controls the sex of an individual is called as the allosome, also called as the allosome, also called as idiochromosome. What is allosome? Allo means different, soma means body. So a chromosome which is different from all other chromosomes, because all other chromosomes are called as autosome. So autosomes are those chromosomes which control the other characters but not the sex determination, are called as the autosomes. And the chromosome which controls the sex may have more genes but controls the sex is called as the sex chromosome, allosome or idiosome. And remember, in the case of the humans, or you can say many organisms, Y chromosome is called as the androsome, andro androsome. So now the next main thing here is, in 1891 Hanking discovered the X body. He was a German scientist. He observed the X chromosomes during spermatogenesis in the frog as well as in the fireflies, and he called it as the X body. And then in 1902, this is explained in NCERT, so please remember, in 1902 Y chromosome, or you can say Y body, was discovered by Stevens. So these 2 points you have to remember: who discovered the X body and who discovered the Y body. Now the next point is, according to this concept, parents can be homogametic or heterogametic. Now what is homogametic and heterogametic? So on the basis of whether the sex chromosome or the allosome is of similar type or dissimilar type, two types of gametes are possible by parents. Please remember, all autosomes will be same. The only difference that can come is in the sex chromosome. So if the parent prepares two types of gamete, because there are two types of sex chromosomes present, then the parent would be called as heterogametic. And if the parent prepares same type of gametes on the basis of the sex chromosome, the parent would be called as homogametic. So now then homogametic can be again of two types. There are males that could be homogametic, or there can be females who can be homogametic. And in the same manner, if males are homogametic, then remember, in the same organism females will be heterogametic. And if the female is homogametic in the organisms, then in the case of such category, males are going to be heterogametic. Let's start with the first one. So the first method is the heterogametic, in which the males carry the XY chromosome. So we know the concept of man, that man sex chromosome or the autosomes are XY and female sex chromosomes are XX. So in the case of the human female, then we have the fruit flies. The females are XX and the males are XY. Whether it is man or it is fruit fly, males will be always XY. So now two types of gametes are possible here. Half of the gamete will carry X and half of the gametes will carry Y. Remember, a gamete which carries X would be, or a sperm which carries X is called as gynosperm. and a sperm which carries Y is called as the androsperm. So if a gynosperm fertilizes the all the egg will carry X only because homozygous situation. So if a gynosperm fertilizes the egg, it will lead to the formation of a female. If a androsperm fertilizes the egg, it will lead to the formation of a male individual. So in this situation, what we have understood is that heterogamety, male heterogamety is a type is the type of sex determination in which two types of gametes on the basis of the sex chromosome are formed by the male parent. So it is present in man, it is present also in the fruit fly. Remember those conditions where males are heterogametic, in those conditions female would be homogametic. Now let's understand one more method, that is in the case of roundworms. That is in the case of the roundworms, or in the case of some insects like grasshoppers are there, cockroaches are there. So remember roundworms, grasshoppers, cockroaches, they will have male heterogamety but different from this. Now in the case of cockroaches, roundworms and grasshoppers, the heterogamety is of different type, but it is a male heterogamety. And the difference is, but in the case of man and fruit fly, the male prepare two types of gametes, one carry X, the other carry Y. But in the case of grasshopper, roundworm and cockroaches, male prepares two type of gametes on the basis of their sex determination. One will carry X chromosome and the other one will carry no chromosome. So that means no sex chromosome. One will carry the X chromosome, other one will carry no sex chromosome. That means all autosomes are normal. The only difference is but one sperm will carry an X and other will carry nothing. So now in the case of female, it will be again same situation, XX. So when a sperm carrying X chromosome fertilizes the egg carrying X chromosome, a female is there. But if a no sex chromosome carrying sperm fertilizes the egg, it will lead to the formation of male again. So males in this category are XO and females are XX.